This explainer is your ultimate, fun-filled, strategic playbook for conquering the Paper 6 exam totally stress-free. We're going to treat this prep just like unlocking power-ups in a game, giving you the exact strategies you need to maximize your score with minimal panic.
4. I literally want you to burn this number into your memory. Why? Because your ultimate boss fight goal for this exam is to lose no more than 4 marks. If you can protect your score and keep your point losses to 4 or fewer, you are right on target to secure that coveted A star. And I promise you, with the tricks we're about to cover, hitting this target is incredibly achievable.
To get us there, here's our roadmap for today. Cracking the Paper 6 code, mastering drawings and graphs, acing the investigation question, essential biology theory, and finally, pro tips for perfection.
Okay, let's dive right into Section 1, Cracking the Paper 6 code.
Look, the absolute most direct way to understand the underlying code of this test is to just grind through the available past papers. There are exactly 15 of them ranging from 2023 to 2025, and you want to solve as many of these as physically possible.
Why?
Well, because the formats, the questions, and the expected answers are highly repetitive. By the time you sit down for the real thing, it's going to feel like you've already taken the test a dozen times. Say goodbye to anxiety and hello to muscle memory.
Moving right along to Section 2, Mastering Drawings and Graphs. You can easily secure 4 free marks just by knowing the strict rules of biological drawing. A winning drawing uses large, crisp, clear outlines. There is absolutely no messy, artistic sketching, and definitely no shading. It's not art class, right? You must count and draw all the specific details, like the exact number of layers or spines. But most importantly, keep the exact proportions. If the specimen is narrower on one side, your drawing must be narrower on that exact same side.
And this right here brilliantly illustrates the golden rule of creating data tables. Whenever you're asked to draw a table from raw data, like say timing how long it takes different sized agar cubes to change color, your independent variable, the factor you are changing, always goes in the left column. The dependent variable, which you're actually measuring, always goes in the right column. And remember, you must include the correct units directly in the headings, not next to every single number down the list.
Graphs are another spot where points are practically handed to you if you just follow the cheat codes. If you're asked to draw a bar chart, make sure your bars are of completely equal width and always leave a gap between them. Always label your axes and include your units. If you're drawing a line graph, you need to plot your points super accurately and join them point to point using a ruler. No freehand curves unless they specifically ask for it.
All right, let's tackle Section 3, Acing the investigation question. This is the big one.
Here is your absolute, foolproof, 5-step combo to ace this 6-mark boss fight every single time. First, identify your independent variable. What are you going to change? Second, your dependent variable. What exactly are you going to measure and how? Third, list 2 control variables to keep the test fair, like using a thermostatically controlled water bath for temperature. Fourth, state that you'll repeat the experiment, usually 5 times, and calculate an average. And fifth, even if they don't ask for it, write down a safety measure. Just write it out step by step, and you're golden.
Let's look at an example from a past paper involving protein digestion. They coated a film in gelatin, which is a protein, and tested how long it took different temperatures of the protease enzyme to turn the film transparent. A massive trick here is something called equilibration. This basically means you put your enzyme and your gelatin substrate in the water bath separately and wait about 5 minutes before mixing them. You do this so they both reach the exact same temperature before the reaction even starts. It is a brilliant little detail to include in your method.
Another phenomenal detail to drop in when you're investigating enzymes is the use of a pH buffer. As proteins break down into amino acids, the pH of the solution naturally changes, which can totally mess up your enzyme. A pH buffer is a crucial substance you add to maintain a constant pH throughout the whole reaction. Mentioning a buffer is an amazing control variable to drop into your answer to easily secure those marks.
Okay, Section 4, Essential Biology Theory. Let's see what you need in your toolkit.
Let's load up your inventory with a rapid-fire theory hit list. You absolutely must know how to identify insects, arachnids, and monocot versus dicot plants. You need to understand the effect of different concentrations on osmosis in plant tissues. You gotta know how to analyze the effect of environmental factors, things like light intensity on photosynthesis or temperature on the transpiration rate. Be ready to label leaf and flower structures, identify xylem and phloem, and measure a pulse rate. Equip these, and honestly, nothing will surprise you.
And listen closely to this one, because food tests are a must in almost every single Paper 6 exam. You need them completely memorized. For example, if you're testing for proteins, you add Biuret indicator without heating, and if protein is present, it turns a violet or purple color. If you're testing for vitamin C, you use the DCPIP indicator. When you add the juice, the DCPIP will change from its starting blue color to completely colorless. Memorize the indicator names and the exact color changes. It's an easy win.
Finally, Section 5, Pro Tips for Perfection. Let's get those last few crucial points. So a huge point here is how you handle comparison questions. It's incredibly common to be given two images and asked to compare them. The golden rule, your points of comparison must only be taken from the visual evidence right in front of you. Do not rely on unseen textbook knowledge. If you know a biological fact about the specimen that isn't clearly visible in the photograph, do not write it down. It literally won't count. Just stick to what you can actually see.
Next up is this magic list of experiment improvements. Whenever the exam asks how you could improve an investigation, just pull straight from this list. First, always do repeats. If they didn't repeat it, say they should. Second, control the temperature properly by using a thermostatically controlled water bath. Third, upgrade the equipment. If judging a color change is subjective and difficult with just your naked eye, suggest using a piece of equipment called a colorimeter, or at the very least, placing a white background behind the test tube.
You're also going to have to do some math, so lock in these essential hacks. To calculate percentage change, take the initial value, subtract the final value, divide that by the initial starting value, and then multiply by 100. If you're asked for a rate, simply divide the change by the units of time. If they want the surface area to volume ratio, make sure you break the numbers down to their absolute simplest ratio, like 1 to 10. And finally, for magnification, remember the formula. The actual size equals the image size divided by the magnification. Take your time with these.
Here is literally one of my favorite, easiest tricks in the entire playbook. Always write a safety step. Even if the question doesn't specifically ask for it, just throw it in. If they're handling chemicals, mention wearing goggles or gloves. If step 2 involves cutting a potato or a plant stem, state that they should use a kitchen cutting board or ensure they cut away from themselves. It is a completely free, easy mark that you can grab just by being cautious. So, we've cracked the code, outlined the magic steps, and loaded up your theory inventory. You are more than ready to totally crush this exam. But think beyond the test for a second. If you can systematically deconstruct a complex biology exam like this, what other complex systems in the real world are you ready to hack, decode, and completely master next? Keep practicing, trust the process, and I'll see you next time.